All right, let's talk now about blood groups, also known as blood types, and transfusions. Um, there's a little historical stuff here about early blood transfusions using lamb blood, um, and in the 1800s, human blood being used with unpredictable results. Um, but Carl Landsteiner is the person who is credited with figuring out the different blood types. In 1910, he identification of the ABO blood antigen gene explained these blood type incompatibilities. Today, there are 35 blood types distinguished by proteins on the surfaces of the red blood cells. And these can be determined by proteins or by the genes that code for them. These discoveries led to development of procedures for typing blood. Safe transfusions of whole blood depend on two blood tests, type and cross match. First, the recipient's ABO blood type and RH status, which we'll be talking about here just in a few minutes. Cross match is done of the recipient's serum with a small amount of the donor red blood cells that have the same ABO type and RH status as the recipient. Compatibility is determined by examining the mixture under a microscope for agglutination, which is the clumping or gluing together of red blood cells. So the explanation for this lies in antigens and antibodies. So an antigen is any molecule that will trigger an immune response, which is the body's reaction to invasion by foreign substances or organisms. When the immune system encounters an antigen that's not found on the body's own cells, it will respond by synthesizing antibodies against the antigen. So antigens, molecules, any molecule that will trigger an immune response. Cells have these molecules, which a lot of different possibilities. Sometimes they're sugars, sometimes they're proteins, sometimes they're a combination, they're glycoproteins. But in any event, all your cells are going to have markers on the outside that allow your body's immune system to recognize them as part of yourself. And this includes red blood cells, which have some different types of antigens that we'll be looking at here in just a minute. So an antibody is something that works against a particular antigen. All right, so in a transfusion reaction, the antigens are called agglutinogens. They will generate or give rise to agglutination. Those are found on the surface of the red blood cells, and they will react with antibodies or agglutinins that are found in the plasma of the recipient. And that can result in agglutination or gluing together of red blood cells. Now, note the distinction. This is not agglutination, is not clotting. There is no fibrin involved. And remember that fibrin was the key to forming a clot. This is called agglutination, and the antibodies will act like glue to stick the red blood cells together. So a mismatched blood transfusion will quickly produce telltale signs of agglutination: anxiety, breathing difficulty, facial flushing, headaches, severe pain in the neck, chest, and lumbar area. Red blood cells will burst and release free hemoglobin. Excuse me a minute, got a cough. So this will lead to red blood cells bursting, releasing hemoglobin. That will break down into hemoglobin. That'll lead to bilirubin, which can cause the skin to yellow, as in jaundice. Anyway, only a few of the known antigens found on red blood cell membranes can produce serious transfusion reactions. And these include the antigens of the ABO group and those in the RH group. So by avoiding mixtures of certain types of antigens and antibodies, you can prevent adverse transfusion reactions. So let's first look at the ABO blood group. You're probably familiar with blood types being either A, B, O, or AB, which is a combination of A and B. In fact, this is something that we normally look at in Biology 103 as an example of something called codominance. So hopefully you're already familiar with this. So blood type frequencies in the U.S., Caucasians, typically you'll find about 45% of Caucasians will have type O blood, another 40% will have type A, only 11% with type B, and only 4% with type AB. Type B is more common in African Americans. Type B is less common in American Indians, who are by far 79% type O. Hispanic population, you'll see more type O at 63%, less type A, only 14%, and 20% type B. So just some interesting differences there. Antibodies that affect the ABO groups are present in the plasma about 2 to 8 months after birth as a result of exposure to foods or microorganisms that contain the antigens that are not present on the individual's erythrocytes. So, in other words, if you have type A blood, you will develop antigens against type B blood within a few months after birth. Let's see. Yes, right here's what I wanted you to see. So this is a red blood cell drawing of one. This represents type A blood. And you'll notice that it's got these little, they look sort of like lollipops, don't you think? Now the real thing doesn't, but for this drawing, that's the way they drew them. So these are the type A antigens. These are the markers on the red blood cell that say, I am a type A red blood cell. Now out here in the plasma, what you find are antibodies that are anti-B antibodies. And again, this is just a way to illustrate it. You'll notice that the ends of these have a green portion with a little V-shaped notch. Now that's going to be important. We look over here, and this is type B blood. So we have a red blood cell, but notice that what's on there, the type B antigens that say I am a type B red blood cell, look like little arrows. And conveniently, those little arrows will fit right into these little notches on these anti-B antibodies. On the other hand, if you look at the type anti-A antibodies, they did a poor job with this. These little V-shaped notches here in orange should have been rounded so that this rounded type A antigen over here would fit into it. I guess that is kind of rounded. My eyesight is not great, but it looks kind of like a V to me. I suppose I could zoom in on it. But anyway, so if you have type B blood, you have type B antigens, and you have anti-A antibodies. If type A blood cells are put into your bloodstream, your antibodies will stick to those and stick to other of the red blood cells with type A antigens and glue them together. If you have type A blood and the type B blood cells like these are put into your bloodstream, the little green arrows fit the active sites on these antibodies, and it will glue these red blood cells together. If you have type AB blood, you have both A and B antigens on the surface of your red blood cell, and you don't have either type of antibody. And the fourth option is to have type O blood. You don't have any antigens on your red blood cells. No type A antigens, no type B antigens. You have no antigens, so that's type O. But in your blood plasma, you see that there are both anti-A and anti-B antibodies. So, what if I take some type O blood cells here and give them in a transfusion to someone with type A blood? Is there anything on the surface of this cell that these anti-B antibodies could latch onto to glue them together? The answer is no. There are no antigens here. What if I give this type O blood cell to someone with type B blood? Are there any antigens on here that these anti-A antibodies could latch onto? No, there aren't. Could I give type O blood here to a person with type AB blood? There are no antigens to be glued together, and there are no antibodies to stick to them. So you could give type O to someone with type AB. Let's see. We can give type O blood to someone with type A, or type B, or type AB. So type O is referred to as the universal donor. More specifically, type O negative, but more about that a little bit later. What about these blood cells? Could I give them to someone with type A blood? No, because these anti-B antibodies would stick to these type B antigens. So if you have type AB blood, you could receive type A blood cells because you don't have antibodies to glue them together. You could receive type B blood cells because you don't have the antibodies to glue them together. You could receive type O blood cells, and the cells do get separated from the plasma, and the antibodies are in the plasma. So I could give you type O red blood cells if you had type AB blood, and there would be no problem. So AB is the universal recipient, O is the universal donor. All right. Now there are also, and I don't remember if there's a good illustration of this. If we move on down here. And all of this is really talking about what I've just mentioned, universal donor, universal recipient. Where is, here we go, the RH blood group. So when you talk about blood being positive or negative, you mean that it is either RH positive or RH negative. The RH blood group was named after the rhesus monkey. Notice how that's spelled, R-H-E-S-U-S, rhesus monkey. So that's where the RH comes from. This is where we first studied it, was in rhesus monkeys. In humans, this group includes several different RH antigens. The most prevalent, the main one of these, is called antigen D, which is a protein located, it is a transmembrane protein, which means part of the protein is on the inside of the cell membrane, part of it passes through to the outside of the cell membrane. If antigen D or the other RH antigens are present on the red blood cell membranes, the blood is said to be RH positive. If you don't have those RH antigens, then you're said to be RH negative. About 15% of Caucasians and 5% of African Americans in the U.S. are RH negative. So most people are RH positive. And this is something that you inherit. You get a gene to give you RH positive or not. The anti-RH antibodies form, now remember, anti-A and anti-B antibodies form within a few months after you're born. But anti-RH antibodies will only form in an RH negative individual in response to the presence of red blood cells with RH antigens. So if you're Rh negative and you never get an Rh positive transfusion, you will never develop antibodies against Rh positive blood. But what if you do? So if you're Rh negative, you could get an Rh positive transfusion once. You would not have the antibodies to glue those cells together, but your immune system would be introduced to Rh positive blood, so to speak, and it would produce Rh, anti-Rh antibodies for future use. So then if you got a second transfusion of positive blood, you would glue those cells together and it could kill you. Another condition where this happens, a similar situation of Rh incompatibility, can arise when an Rh negative woman is pregnant with an Rh positive fetus. And that is very possible. If the father's Rh positive, the baby could be Rh positive. The first pregnancy with an Rh positive fetus would probably be uneventful, but if at the time of this infant's birth the placental membranes that separate mom's blood from the baby's blood tear and some of the infant's Rh positive blood gets into the mother's bloodstream, they can stimulate the mother's immune system to produce anti-Rh antibodies. It could also happen if the pregnancy ends before the birth, or fetal cells enter maternal circulation during an invasive procedure like an amniocentesis. So if a woman who's already developed anti-Rh antibodies becomes pregnant with a second Rh positive fetus, those antibodies are actually small enough to cross through the placental membrane and cause the baby's own blood cells to be glued together. The condition is called erythroblastosis fetalis, also known as hemolytic disease of the newborn. Now we prevent this with a shot. It is called, here it is, RhoGAM. An Rh negative woman who might be carrying an Rh positive fetus will get an injection of the drug RhoGAM at week 28 of her pregnancy and after delivery of an Rh positive baby. So this is, RhoGAM is anti-Rh antibodies. So what they will do, I like to use an analogy. If there's a small country that gets invaded by a larger country, but it doesn't have an army, it could build an army of its own. That's not the best analogy, but however, if another country sends an army to defend that smaller country, the smaller country doesn't need to make its own army. And then once the invading country has been pushed out, the larger country that sent the army in can withdraw. And that way, there's not an army in that country. Okay, I hope that made some sense. So if you give an Rh negative woman anti-Rh antibodies, they're not going to stay in her body from then on, but her body will not have to produce any. So the next time she has an Rh positive baby, she will not have her own antibodies to work against it. I hope that makes sense. Anyway, that is it. And if I didn't explain that clearly enough, y'all tell me and I'll come back and try to clarify it and make another video. I don't think that there is, no. This would be something where an animation would be really helpful. I need to try to remember to suggest that to the folks at McGraw-Hill. All right, so that's it for the chapter on blood.